Hi, this is Dr. Xu teaching design of experiments for engineering research. In this video, I will teach how to perform ANOVA analysis of the experimental data by hand calculation. This will help you to understand the detailed procedures in the analysis of variance. We will use these three groups of data. You can think of them came from strength test results for material with added different levels of a chemical. And we would like to know if the concentration of the chemical has impact on the material strength. The null hypothesis is all the means for differently treated materials are the same. And the alternative hypothesis is that at least one mean is different. So if we reject the null hypothesis, that means the concentration of the chemical does have impact on the material strength. We use the significance level of 0.05. We firstly need to calculate the mean square between groups. This is basically the between groups sample standard deviation. It is the sum of squares between groups divided by the degree of freedom, which is the number of groups minus 1. In our example, it is 3 minus 1 equals 2. To calculate the sum of squares between groups, firstly we calculate the mean for each group. So the mean for group 1 is 4, for group 2 is 8, and for group 3 is 13. Then we calculate the mean for all the 15 observations, and the result is 8.3. Next, we subtract the global mean from the mean of each group. So 4 minus 8.3, 8 minus 8.3, and 13 minus 8.3. We get the result here, here, and here. Next, we take the square of the above results and multiply by the sample numbers in each group. So each group has 5 samples. So 5 multiply the square of the result here. And we will do the same thing for the second data group and the third data group. The results are here. Lastly, we sum the results to get the sum of squares between groups. So this number plus this number plus this number, and the final result is 203.3. Therefore, the mean squares between groups is 203.3 divided by 2 equals to 101.667. The above calculation can be expressed as mathematical notation shown here. After calculating the mean square between groups, which is related to the variation between groups, next we will calculate the mean square error. This is basically the within groups sample standard deviation. We use this to quantify the variation within each group or within each treatment. It is calculated by dividing the sum of the squared error by the number of observations minus the number of groups. So this is the degree of freedom for the mean square error. In this example, it is 15 minus 3 equals to 12. To calculate the sum of squared error, we firstly subtract the mean for the group from the observations within that group. So the mean for the first group is 4, and we subtract it from each of the values within this group. So 2 minus 4, 3 minus 4, and we do the same thing for the other observations. We will repeat the same procedure for this group and for this group. Then we take the result here, square it, and sum up the squared results. For the first group, we got 22. We will repeat the process for the rest of the groups, here and here. And lastly, we sum up the results, so 22 plus 18 plus 14. We get the sum of the squared errors of 54. Finally, the mean square error equals to 54 minus 12 equals 4.5. The sum of squared errors calculation can be expressed as mathematical notations shown here. Another related concept is the total sum of squares. This total sum of squares divided by its corresponding degree of freedom which is the total number of observations minus 1. This is basically the total sample standard deviation. In that, we calculate the mean for all the 15 observations, so the global mean is 8.3. Then subtract it from each individual observations. So subtract 8.3 from each of these individual observations for all the groups. Next, square the results. So we have the result here, we square the result, and we do this for all these results and sum up all of them. So the total of these, and these. Finally, we got the result is 257.3. The total sum of squares calculation can be expressed as mathematical notation shown here. The total sum of squares is related to the total variation of all observations. It has two components: the sum of square between groups and the sum of the squared errors. The sum of square between groups plus the sum of squared errors equals to the total sum of squares. The sum of square between groups is related to the variation caused by different treatment, and the sum of the squared error is related to the variation within groups. So in our example, this value equals to this value plus this value. Basically, if between group variation is larger than within group variation, the variation between the groups are more likely to be caused by the different treatments. Lastly, we calculate the F-score and perform an F statistical analysis. It equals to the mean square between groups divided by the mean square error. Basically, this expression is the between group standard deviation divided by its degree of freedom over the within group standard deviation divided by its degree of freedom. If you recall from the chi-square distribution and F distribution lecture, this is an F distribution that can be used to test if the between group variance and the within group variance are the same. So in our example, it equals to 101.6674 divided by 4.5, and the final value is 22.59. By using this function in Excel, so this is the F value, this is the degree of freedom in the numerator, and this is the degree of freedom in the denominator. We get a very small p-value, much smaller than 0.05. So we reject the null hypothesis and conclude that chemical concentration has strong impact to the mean material strength. If we summarize all the calculated numbers, we have this ANOVA table. We have the source of variation from between groups, within the group, and the total variation. The degree of freedoms are calculated here, and the calculated SSG, SSE, and SST. The mean square value is this number divided by this number, and this number is this number divided by this number. Finally, the F value equals to this number divided by this number. And we can put the corresponding p-value here. You may notice that in the example, each treatment or each group, we have equal number of data. So in each treatment, we have done 5 tests. This case is called the balanced data. This is preferred in designing an experiment with one factor, because first, it is relatively insensitive to small departures from the assumption of equal variance for different treatments. And second, the power of the test is maximized, so we are less likely to make wrong conclusions. The calculation of the ANOVA table is summarized in this slide. For unbalanced data, the calculation of sum of squares between groups and sum of squares total are slightly different, and they are shown in the highlighted box here. The rest of the calculations are the same. There are 3 conditions that we must check on the data before performing ANOVA analysis. The first one is that all the observations should be independent. The second one is the data within each group should be approximately normal. And the last one is that the variability across the groups should be about equal. Another thing we need to know is that the ANOVA only tells if there is difference in the means of different groups or different treatment. It does not tell you which of these groups have different means. To answer that question, we will need to compare the means of each possible pair of groups using a t-statistic. We will not look at this for now. Alright, this is Dr. Xu. Thank you very much for watching. Please comment if you have questions. Subscribe, share, and like the video if you think it is helpful. I will see you in the next video.